This is my final project for the statistical learning course offered at Westminster. Statistical learning focuses on the understanding of the relationships between variables, emphasizing interpretability and how inputs influence outcomes. Machine learning, on the other hand, prioritizes predictive accuracy, often using more complex models, even if the internal workings are less interpretable. The goal of this project is to apply what we learned in the course on both classification and regression models onto a complex data set to generate predictions. I chose to use generated data from the MSPrime package. MSPrime is a simulation tool widely used in genetic research to generate realistic models on how DNA is inherited across generations. In this project, it produced variables such as simulated genetic markers, environmental effects, and trait values, allowing us to study how different factors influence both a continuous trait and a binary disease outcome in a controlled and realistic way. This is the code that generates the data as a CSV file with 8 columns of data. I trained the regression models to predict quant trait, a continuous variable similar to height, blood pressure, or lung capacity, anything that is on a continuous scale, and the classification models on disease status, a discrete binary yes or no variable similar to whether or not you have type 1 or type 2 diabetes, celiac disease, or even color blindness. Before we can train the models, we want to see the data through the EDA. Here's the summary statistics, including the 5-number summary, mean, and standard deviation. Scrolling quickly through this data, we can see the bar charts of my numerical data, which shows us the shape of the data, whether or not it's left or right skewed, and the correlation matrix, which tells us which variables seem correlated with another variable. The count histograms for the categorical data and the correlation matrix between the categorical variables. Then the full correlation matrix with all the variables and the PCA graphs. Then we have our outlier analysis with both box plots and the z-score. These are the classification models I ran, a total of 14 different models provided by the scikit-learn package. 5 were disqualified due to potentially overfitting the data. The models disqualified were KNN, decision trees with Gini and entropy, bagging, and the random forest. Of the qualified models, logistic regression had the best area under the curve and precision. Here's the overall stats of the log regression. An accuracy of 68.6 for predicting whether or not someone has the disease we trained the model to detect. That's noticeably better than guessing, which would be a 50-50. It shows that the information in the dataset contains real signals that can help identify who is at risk. This is interesting because it mirrors how real-world medical risk models work, using patterns in someone's genetic or biological data to estimate the likelihood of having a condition. Similarly, here are the 16 regression models I ran from scikit-learn. They were disqualified due to potentially overfitting. They were all the regression trees on different formats of the data. Of the qualified models, the linear regression on the full data, including the PCAs, was the best. Here's the overall stats of the LR, showing that it captures about 56.7% of the factors that influence the trait that we're predicting for. For real biological traits, getting more than half of the variation explained through measurable factors is quite strong, because traits are usually influenced by a mixture of genetics, environment, and randomness. This makes the result very interesting because it shows that the model is capturing meaningful biological structure in the data, not just random noise. Overall, these results demonstrate how statistical learning tools can uncover relationships, both for predicting disease presence and estimating continuous traits, using only the underlying patterns of the data.